Hi, today I'm going to be talking about the events of DNA replication, and so we can understand how the DNA replicates so it can reach this DNA sequence, which is made of adenine, cytosine, guanine, and thymine, which are connected with either a double hydrogen bond or three hydrogen bonds. Um, Adenine, cytosine, guanine, and thymine are made of deoxyribose sugar group, nitrogen containing base, and phosphate group. And the red thing here, as you can see, is the sugar phosphate backbone, and here is the deoxyribose, which is at each strand, either if it's the strand of adenine and thymine or guanine and cytosine together. And here is the phosphate group, which is over here. So as you can see here, there is a phosphate group, nitrogen base, and deoxyribose. So the deoxyribose is here at each single strand, and the phosphate is here, which is that. Before I start talking about the three main events of DNA replication, which is initiation, elongation, and termination, I'm going to talk about how scientists discovered which type of the DNA models that were proposed back then was the correct one. So there were three proposed DNA models. The first one was the conservative model, and it proposed that the replication involved formation of two new daughter strands from the parent templates. So the two new daughter strands are joining together to create a new double helix. And the two original strands, which are the parent strands, would then reform into a parent molecule. And the second one was the semi-conservative model. So the semi-conservative model proposed that each new molecule of DNA would contain one strand of the original complementary DNA molecule and one parent strand. And that would conserve half of the strand of the original molecule, and it would have the other half, which is the new one. And the third one was the dispersive model. So the dispersive model proposed that the parental DNA molecules were broken into fragments, and that both strands of DNA in each of the daughter molecules were made up of an assortment of parental and new strands. So here, as you can see, for the conservative model, it's only one parent strand and one daughter strand. And for the semi-conservative model, one strand is parent and daughter mixed together, and one of them is only parent. And for the dispersive model, it's a mix of both small fragments from parent and daughter. But until that time, no one concluded no one really concluded which type of model was the correct one for the DNA replication until Matthew Meselson and Franklin Stahl decided to use two different types of isotopes, and which were nitrogen-14 and nitrogen-15. Nitrogen-14 was basically called as the light nitrogen, and nitrogen-15 was known as the heavy nitrogen. So why did they use nitrogen instead of using any other element? And here we have an example from section 1 that we learned. When Alfred Hershey and Martha Chase, they used 32P as an isotope to trace DNA from in the T2 bacteriophage strain of virus, when they were trying to figure out if proteins or DNA were the ones who contained the genetic material. Well, the reason why they used nitrogen is because nitrogen is an essential component of DNA. So it would be incorporated into any new daughter cells that were synthesized to the cell. Also, the fact that there's N14, which is light, and N15, which is heavy, makes it easier for the scientists to separate the different DNA strands according to how much of each isotope was present. So how did the experiment go? They first put the bacteria in an N15 medium, and then they transferred it to an N14 medium, which is the light nitrogen. So when they transferred it to an N14 medium, they saw that the DNA sample formed a single band. And the density of that single band was between N15 and N14. So the scientists concluded that the DNA was composed of one strand labeled with N14 and one strand labeled with N15. And that's why they ruled out the conservative model. And remember here, as we saw earlier, the conservative model, it had only one parent strand and one daughter cell strand. But when they put it in the N14 for the first round, it showed a mix of both, and that's why they took away the conservative model. For the second round of replication, the DNA sample separated into two different bands. And one band was only N14 and the other band was only N15. So then in the second round, the DNA sample separated into two different bands. One band was N14 and the other band was N15. And that's why scientists ruled out the dispersive model. And remember here, the dispersive model was small fragments of a mix of the parent strand and the daughter strand. And that's why they ruled it out, because it wasn't like that. It was one strand of the N15 and one strand of the N14. So this experiment supported that the semi-conservative model was the correct model of DNA replication, because we have one parent strand and one daughter cell strand. So now that we learned about how scientists concluded that the semi-conservative model was the correct model for DNA replication, let's see the 3 steps of DNA replication. So the first step is initiation phase, which is a portion of the DNA double helix when it unwinds to expose the bases for new base pairing. Then we have elongation, which is when 2 new strands of DNA are assembled using the parent DNA as a template. And one of these strands are parent DNA and the other strand is a daughter DNA. And lastly, we have termination phase, which is when the replication process is complete and the 2 new DNA molecules separate from each other. Since we talked about each step of the DNA replication and we have an idea about what happens in each one of them, let's dig deeper and see what happens in every single step. So in initiation, the replication starts at a specific nucleotide sequence called the origin of replication. And that's where initiator proteins bind to the DNA and begin the process of unwinding the double helix. One group of proteins that's involved in the unwinding process is helicase, and that cleaves the hydrogen bonds that link the complementary bases between strands together. So helicase weakens these hydrogen bonds right here. So these hydrogen bonds that link together adenine, thymine, guanine, and cytosine, the helicase, it weakens these hydrogen bonds until it breaks. And then we have the single strand binding proteins, which help to stabilize the newly unwound single strands. And those single strands have a tendency to reform into a double helix if they're not checked. So once the helicase weakens the hydrogen bonds, each side becomes a single strand. They're no longer connected together. And we also have the topoisomerase 2 enzyme that helps to relieve the strain on the double helix sections ahead of the replication forks, which result from the unwinding process. Initiation creates a shape called replication bubble with two Y-shaped regions at the end of each of these area. And the Y-shaped are called replication fork. So if we look here, we're gonna see that the origin replication started here, and then you can see that helicase started unwinding the hydrogen bonds between the strands. So then it started separating and it started becoming a single strand. And here, that's when the replication fork starts to form. So here you can see that's the replication bubble, and that's the replication fork, the Y-shaped area. So now that we learned about initiation phase, let's talk about elongation. So in elongation, it synthesizes new DNA strands by joining individual nucleotides together. DNA polymerase 3 is the enzyme that catalyzes the addition of new nucleotides one at a time, and it creates a strand of DNA that's complementary to a parental strand. DNA polymerase 3 only attaches new nucleotides to the free 3-hydroxyl end, and it only synthesizes a new strand from a parent strand in the 5-3 direction towards the replication fork. So when the double-stranded DNA is separated, there would be two types of strands, the leading strand and the lagging strand. For the leading strand, the synthesis of the new DNA happens once and only once. However, in the lagging strand, the synthesis of new DNA happens in short segments and in a discontinuous manner. So these short segments of DNA are named Okazaki fragments. The Okazaki fragments are short DNA fragments that are generated during the synthesis of lagging strand in DNA replication. So how does the lagging strand synthesize new DNA? It starts with RNA primers, which are short fragments that are used to start the DNA replication. They are synthesized by an enzyme called primase. Once these primers are in place, the DNA polymerase extends the strand by adding new nucleotides to the 3-hydroxyl end of the primer. Once each primer is added, a new DNA fragment is generated from the end of each primer, which results in the synthesis of the Okazaki fragments and which are a series of short fragments of DNA that each begin this section of RNA. Eventually, another DNA polymerase enzyme, which is DNA polymerase enzyme 1, removes the RNA primer and fills in the space by extending the neighboring DNA fragment. The Okazaki fragments are then joined together by the enzyme DNA ligase. And lastly, we have termination, and that's when the replication fork progresses along the replication DNA. Only a very short region of DNA is found in a single-stranded form. As soon as the newly formed strands are complete, they rewind automatically into their chemically stable double helical structure. So by termination, the shape of the DNA sequence is back to this, and they're not single-stranded anymore. So termination occurs upon completion of the new DNA strands and the two new DNA molecules separate from each other.